CanCOVID is an open science collective dedicated to rapidly mobilizing and sharing knowledge to help inform Canada's COVID-19 response. To learn more about us or how to join our community, you can visit our website at cancovid.ca.
Well, welcome everyone. Thank you all for joining. Today we are hosting our CanCOVID workshop on therapeutic advances, and we have 5 very interesting talks for you today, and also a Q&A session at the end where we will look forward to a respectful discussion. And I just want to briefly introduce our moderator for today, who is Dr. Peter Jüni, who is the Network Science Advisor for CanCOVID, as well as the Scientific Director for Ontario, the COVID-19 Science Advisory Table. So I will not take up much time today as we have lots on the docket, but I'll pass this over to Dr. Peter Jüni. Thanks.
Thanks a lot, Rosa. Hi, everybody. So I'm looking forward to this session today. We will start with Emily McDonald, who will give us an overview on how treatments have evolved for outpatients, ward patients, and in critical care, followed by talks related to several of the trials with a lot of Canadian involvements and the WHO prospective meta-analysis initiative. So I'm handing over to you, Emily, first. You're an associate professor of medicine at McGill. You have been involved in quite a lot of the trials that I have been involved too. And you know just the 101 of treatments and how it has evolved for the various subgroups of patients. Handing over to you, Emily.
Hi, thanks, Peter. Thanks, everybody, for attending today. Very excited to get things started. There's lots to go over. So I will just start with my disclosure. So I do have public funding, and I've been the PI and co-PI on several COVID-19 treatment trials, several of which we'll be discussing today. I'm a member of INESS and of the Canadian Task Force, but the views that I present today are my own. There's so many interesting treatments and studies to talk about, and a lot of the landmark trials will be presented today by the PIs. Won't be able to go into every treatment and all of the evidence behind every different therapeutic that's out there. It's evolving so quickly because of time constraints. I'll try and focus on some high yield topics. So for outpatient, we'll go over the variety of different medications that have been tested. Some of them have been eliminated. Some of them we're gathering more information, and I think we'll have an answer in the next few months. And then for inpatient and critical care, just reviewing a little bit about dexamethasone, remdesivir, and the various other treatments that we can use for patients once they're hospitalized. And the different treatments, a lot of them, of course, are based on the needs for oxygen or ventilatory support. So in the beginning, there was hydroxychloroquine. I was asked to present an overview of how things have evolved over time. So several different randomized control trials, including pre-exposure prophylaxis, early treatment, and also post-exposure prophylaxis showed no significant difference over placebo. So for post-exposure prophylaxis, a well-powered trial that showed no difference. For early treatment, a trial that was repowered because the rate of hospitalization was so low that it was repowered for symptom resolution, which showed no difference. And then going all the way back to very early in the disease, which is pre-exposure prophylaxis, a large trial of over 1,000 healthcare workers that showed no difference taking hydroxychloroquine once or twice a week. So that was really, to me, where things started. These were some of the first randomized control trials that were published at the beginning of the pandemic. I'm moving on to a somewhat contentious issue now, which is the drug ivermectin. So this is it's a beef farm in Quebec, outside of Sherbrooke. This is ivermectin, which is used on the farm in order to deworm the cows. We also use ivermectin for other treatments in humans, so as an antiparasitic, particularly for the treatment or prophylaxis against reactivation of strongyloides. We'll only show this one trial for ivermectin just to say that really no difference in time to resolution. This was a placebo-controlled randomized trial that was published in JAMA. Hazard ratio for symptom resolution, really no difference. And then an absolute difference and reduction of symptoms that was not statistically significant. So going anywhere from a 4-day reduction to all the way to increasing symptom duration to 2 days. So really not recommended, and certainly not recommended to use the veterinary formulation. And there have actually been Health Canada and FDA warnings because this is in fact taking place. Next, I'm going to talk about azithromycin, also probably not good, widely prescribed. This is a trial that was published in JAMA, also a randomized controlled trial, small, placebo-controlled, where participants were randomized to a large dose of azithromycin, 1.2 grams versus placebo. And this study was also looking at symptom resolution. And so really, no difference in symptom resolution between people who did and did not receive azithromycin as compared to placebo. So, you can see that over time, what's been happening is a lot of the treatments that were kind of being given on spec, we're starting to gain randomized controlled trial evidence. And so I think what we're starting to do is crossing these off the list of things that we're giving to outpatients. So we shouldn't be giving hydroxychloroquine, we shouldn't be giving ivermectin, we shouldn't be giving azithromycin. So we're going down the list here. Colchicine. So moving into the realm of maybe it's a little bit good. So colchicine was one of the largest randomized control trials, certainly the largest randomized control trial in outpatients in Canada. Thousands of patients were recruited. It didn't reach its primary efficacy endpoint of reducing hospitalization and death. In the beginning, when PCR was harder to obtain, the study included participants who didn't have PCR confirmed disease. When the study investigators looked at the subset of patients who were PCR confirmed, there was a slight reduction in hospitalization. It's been reported as a very modest effect size. So about a number needed to treat of 70. And there was a significant increase in gastrointestinal adverse events in the colchicine arm when it was compared to usual care. There were subgroups in the colchicine trial, so colcorona, where the number needed to treat was less. So particularly older men with diabetes or hypertension, the number needed to treat was more down around 40 to 50. And, this modest effect size may be more relevant depending on how great a crisis your country or your jurisdiction is facing and whether or not you have the means to provide hospitalization to outpatients. So I think something to keep in mind, but most jurisdictions have said that a confirmatory trial is needed. Next topic I wanted to talk about are inhaled corticosteroids. So this is a topic that I have some experience with. The third trial in the list here is the CONTAIN trial, which was run out of McGill. So Nicole Easer and myself, Nicole is a respirologist, we ran the CONTAIN trial. And so I'll talk to you a little bit about some of the trials that have been published in the meantime, and another trial run by Covis Pharmaceuticals, which was published as a preprint just recently. So STOIC was kind of the original study. This was published in Lancet Respiratory Medicine, pretty small trial, about 150 people, randomized 1-to-1. And just to remember that it wasn't placebo controlled, it was open label. This was COVID-19, the primary outcome was COVID-19 urgent care visits or hospitalizations. And you can see that in the budesonide arm that occurred in only 1 participant and 10 participants in the usual care arm. So that was statistically significant with a number needed to treat of 8, so quite interesting. Next came the principal trial, which was a much larger trial. It came out as a preprint quite a while ago, and then was published more recently, peer-reviewed in the Lancet. This study looked at inhaled budesonide. You can see close to 800 people received inhaled budesonide compared to other treatments. It was randomized, but again, just want to draw your attention to the open label nature, so not placebo controlled. I'm mentioning this because there is a good literature on the placebo effect of inhalers, and that this is a medication that, if at all possible in a trial, you would try to placebo control. So the principal trial showed a benefit in time to recovery. So they used a Bayesian credible interval that looked like there was improvement of 2.9, so about 3 days in the budesonide group compared to usual care. And there was a hazard ratio of 1.21 with a probability of superiority greater than 0.999 to meet the pre-specified superiority threshold of 0.99. So high probability of superiority. And similarly, didn't quite meet statistical significance, but a high probability of superiority with respect to hospital admission or death as the outcome. So also looking quite promising, but with methodologic limitations. This was our trial out of McGill. We completed enrollment a couple of months ago. We were powered to show a decrease in symptoms among non-hospitalized patients at day 14, and we actually gave inhaled and intranasal ciclesonide at quite high doses. Ciclesonide is interesting because it's one of the inhaled corticosteroids that has also shown an antiviral effect in vitro. And our second objective was to look at hospitalizations. There were no deaths in our trial. In general, the outpatient trials have very, very few deaths, and the rate of hospitalization in trials recruiting in Canada for outpatients and in the US has been quite low, so somewhere around 5%. So a lot of these trials end up being powered for symptom resolution. Our trial is under review. We were placebo controlled, so those results should be published pretty soon, I would imagine, in the next few weeks, hopefully. And then I will circle back to this trial, which was run by Covis Pharmaceuticals, who makes ciclesonide. Covis donated the drug for our trial, but they weren't involved in the trial design or conduct or write-up of the study, but they did do their own trial, which was just published preprint in medRxiv. Theirs is also a placebo-controlled trial. So this is the first one to be kind of put out there in the scientific domain that was placebo-controlled. There were 200 to ciclesonide and 200 received placebo. There was no difference in the primary efficacy endpoint of time to alleviation of COVID-19 related symptoms. So when they placebo controlled their trial, there was no impact on symptoms. Interestingly, they showed a decrease in hospitalizations, but there were so few, it's very fragile. So it was something on the order of like 1 versus 5. So we'll wait to see the final publication of that and look forward to the results of CONTAIN when they're published. I think that the placebo effect is quite real with inhalers and that we need to keep that in mind. Next on to the drug Fluvoxamine. So this is an SSRI with many different potential mechanisms against COVID-19. I will say stay tuned for Professor Ed Mills' talk, which is coming up. There have been a couple of other trials as well that have been published. So Stop COVID-1 and Stop COVID-2. Stop COVID-1, very positive, showed a reduction in hospitalization, and Stop COVID-2, which we participated in at McGill, completed and has not yet been published. And then I think Ed is going to talk to you a bit more about the Together trial. On to monoclonal antibodies. So know your variants. For people, these are for outpatients who are at high risk of progression to severe disease. So the usual risk factors that you're familiar with: older age, immune suppression, obesity, cardiovascular disease, hypertension. In the US, monoclonal antibodies are also being given as post-exposure prophylaxis in addition to treatment. They are prioritized for people who are unvaccinated or for partially vaccinated people who are at risk of progression despite their vaccination. In Canada, you might be familiar with bamlanivimab, which we have quite a few doses. Health Canada authorized the single monoclonal antibody for use in November. In the US, around springtime, they stopped recommending its use as a single agent because there was signs of resistance among different variants. So some variants were no longer showing an effect. And then Health Canada released a safety alert for the single monoclonal antibody that there was a risk of failure, and that was released in April 2021. In the US, they are using bamlanivimab plus etesivimab as an IV infusion, where the combined frequency of resistant variants is low. There's also casirivimab plus indevimab available as IV or sub-Q, and this was granted Health Canada authorization in June. And then there's sotrovimab, which was also granted Health Canada authorization in July. And if you've been following this story, I think definitely not being widely used in Canada. Some will say underused, and we do have access to the single monoclonal antibody. Then you would really need to know the variant that you're treating in that case. Moving on to patients who have been hospitalized. So you have patients who are hospitalized who aren't requiring critical care. Maybe they're hospitalized and not needing oxygen, but they're at risk of progression. You have patients who are on some level of oxygen, and then people who are requiring non-invasive support, high flow oxygen, non-invasive ventilation, and then finally you have patients who are intubated and on ECMO. And so, you know, what treatments you give is really divided up. People are classified according to whether or not they need critical care and their level of oxygen and ventilatory support. And that is pretty much how we define who gets what treatment. You know, the mainstay of therapy for people requiring oxygen and who are requiring more ventilatory support is really dexamethasone. So dexamethasone, good. This was the recovery trial that was published in the New England, which compared dexamethasone to usual care, big trial, over 6,000 patients, 482 in the dex group and 1,000 patients in the usual care group. And we can see that as your level of ventilatory support went up, that you had people who were receiving oxygen had a decreased rate of deterioration and death when they received dexamethasone. For people who were receiving no ventilatory support, this wasn't the case. So a very nice kind of pharmacoepi study where you can see a sort of dose response depending on the severity of the disease. So next we have remdesivir. Might be good. This is really one that's kind of like gone back and forth in terms of the pendulum. So, you know, in the beginning, we had some studies that showed that it reduced the duration of hospitalization. Then we had the WHO results from Solidarity in March. And now there are more trials that are going to be coming out and that have been published quite recently with remdesivir, some negative, some positive. So, you know, I think that this concept of the living systematic review and meta-analysis that is able to really calculate the sum total of the evidence is very important, will be very relevant for this drug. So I'll just say maybe stay tuned for Dr. Murthy's talk. Next, we have low molecular weight heparin. So can be prescribed to patients who are on the ward, receiving ward-based care, not yet requiring critical care. So we say sometimes good, sometimes bad. So that pretty dichotomous findings from these trials, which Dr. Zarychanski is going to talk to you about today. Finally, we move on to sicker patients. So people who are, you know, having their oxygen delivery through high flow devices or non-invasive ventilation, and then all the way to patients who are requiring intubation, mechanical ventilation, or ECMO. And so again, you can see that in these patients, really the mainstay of therapy is dexamethasone. And you may choose to either give baricitinib or tocilizumab to these patients. Go into a little bit more detail about the who, so which patients might benefit, and the when, the timing, which is pretty relevant. Some people will also give remdesivir in conjunction with dexamethasone, which is more of an expert recommendation and a kind of theoretical concept that maybe dexamethasone will slow the viral clearance. So that's, you know, this is from the NIH, these guidelines, and that's a level B3, which means that there's some trial evidence, but there's methodological limitations, and it's more of an expert recommendation. So we have the IL-6 inhibitors, which have come out as being quite interesting and important. So just talking about the evolution of care, these medications were really being given kind of off-label. Observational data was conflicting. And so, we waited for many months for the randomized control trial evidence. So we have REMAP CAP and recovery. These were two large randomized control trials. There was a mortality benefit for tocilizumab for people who had kind of rapid respiratory decompensation, so hospitalized and very quickly needing high flow oxygen or non-invasive ventilation. I think just to keep in mind that in these studies, most patients concurrently received corticosteroids. So most people would recommend that these are given together. REMAP CAP, some take-home points from that study was really that there was early enrollment of people who had rapid rest decompensation. So on average, people received tocilizumab within 2 days of hospitalization or with 1 day of critical care. So I think timing is really critical, and people who have large oxygen needs and big support for ventilation are more likely to benefit. And then in the recovery trial, just the kind of added criteria that the CRP was elevated in these patients, so CRP over 75. REMAP CAP also posted an update showing that sarilumab is probably non-inferior to tocilizumab. Again, sarilumab in combination with steroids. I think based on the sum totality of evidence, what most people would recommend is that you would attempt to give tocilizumab just because there is more data. But if it's unavailable, then you could consider giving sarilumab. Here is if you want to give some ivermectin, please go ahead. So if your patient's from an endemic area and they're at risk for strongyloides, they're receiving tocilizumab and steroids, we would want to reserve this medication for its known effect, which is to treat or prevent strongyloides in these patients who are at risk of reactivation. Finally, there is some pretty recent data looking at these kinase inhibitors, so baricitinib and tofacitinib, again in combination with dexamethasone. You would want to avoid prescribing these in combination with IL-6 inhibitors. Both are profoundly immunosuppressing. There is some total more evidence for baricitinib than there is for tofacitinib. There's no head-to-head trials, mind you, but the COVE barrier trial had quite a few patients, so 1,500. These are people who had increased inflammatory markers, and it was a placebo-controlled trial. So just I'll draw your attention to decreased all-cause mortality. You can see a hazard ratio that was significant here of 0.57. There's also the ACT-2 trial. These patients didn't receive steroids. Baricitinib in combination with remdesivir decreased time to recovery. So could be a consideration for patients who are for some reason unable to receive corticosteroids, that you could give this combination. And then tofacitinib, there's evidence from a trial called Stop COVID, also placebo-controlled, but much smaller. So about 300 people in this trial also showed decreased all-cause mortality. So just because of the larger trial for baricitinib and then the additional safety data from ACT-2, if you have baricitinib available, then you would probably use that over tofacitinib if you're trying to decide between both at this point. And, the list goes on. It's great. There's so much research that's taking place, but there's, every other week, there's a new monoclonal antibody or monoclonal antibody combination. Use of monoclonal antibodies in hospitalized patients who have, negative baseline serology. There's a really interesting study on anakinra, which is an IL-1 antagonist that was just published in Nature Medicine. Right now, not really made it onto the guidelines in most jurisdictions that I looked into, but this is based on a marker called SUPAR, which is a marker of risk for disease progression. So may not be really readily available in your center, but keep your eye on anakinra, could be interesting. And then there's lots of other things that are being looked at. So GCSF, interferon alpha and beta, TK inhibitors, other Janus kinase inhibitors. And then, there's finally some treatments that we're pretty sure that we're going to recommend not to give. So non-SARS-CoV-2 specific IVIG. And then, when it comes to recovered plasma, there's a wealth of evidence and it's really pretty evenly balanced. So some trials showing improvements, some trials showing no improvement. So a lot of places don't have, may say it's insufficient evidence or that they can't really pronounce based on the balance of the weight of the evidence. So there's a ton of different treatments that are available. We started off with random care. I think we're now giving a lot more care based on randomized trials, which is amazing. If you're looking for some really good resources, Canadian CADITH guidelines are really good. They're updated pretty frequently, and they have a news post that will tell you about the latest treatments. NIH also has a similar approach where they post up new data. There are, of course, provincial guidelines. And so if you're in Ontario, Quebec, there's INS, BC, all provinces have their local guidelines, which are important to take a look at because different provinces have different medications that they have access to. And so that can change from province to province. And then, of course, there's lots of different interesting people to follow on social media. So A.B. Stewart is kind of daily posting when new trials are published, and lots of other interesting people and guidelines to follow on Twitter. And I will end there. Thank you.
Thanks a lot, Emily, for this overview. I'm handing over directly to Ed Mills. You're a professor at the Department of Health Research Methods, Evidence and Impact at McMaster. Ed, I've known you for such a long time, I can't remember when we met first. You will be talking about the Together trial. Handing over to you directly, if I may.
I hope you can see my slides okay.
Yeah, I think it's coming. Great, yeah.
All right, great. Thanks, Peter. Yeah, I think we met in Brazil many years ago. So ironically, I'm going to be talking about Brazil today. This is based on a large platform trial, an adaptive platform trial that we have conducted on early treatment of COVID. Some people may be unaware of some terms that I use here. So adaptive means that there are changes made throughout the trial as emerging evidence becomes convincing that you should change your trial. And then platform trial means essentially that you can evaluate more than one intervention at any time. And I think we've tried to really push the boundary on where trials, what's innovative in trials. And so there has been quite a mix in terms of, the COVID response has gone from case series, like we remember from France in hydroxychloroquine, through to some really very convincing platform trials. Now, in general, although there's been about 3,000 trials planned for COVID, the majority of them are going to end up being useless. The average sample size is about 100 in all trials. And, the good quality trials could have answered this with just one. So for example, looking at trials that investigated hydroxychloroquine alone in a hospitalized setting, there was almost 5,000 patients, that is planned for recruitment, which is over 3 times the total N that was required for the recovery trial that concluded it was not beneficial. So I talk about highly efficient clinical trials. And what do I mean by highly efficient trials? We get this highly active, highly efficient when we think about what was it that changed the course of the pandemic in HIV, for example, because there's a lot of lessons to be learned from HIV in COVID. Key points are know your epidemic. So are the events common enough to save time, sample size, and resources? It's very difficult to do good quality clinical trials in Canada for really any condition. But for COVID, it is very, very difficult because you need to get enough people who are at risk of having the illness and then developing more serious illness. Next issue of it would be, are there plans to make changes during your trial? We do a lot of modeling of possible scenarios. What happens if we have poor adherence, dropouts, completely unexpected events that occur. Event rates within a trial are unlikely to be what you thought they might be at the beginning, particularly in this illness, because it comes in waves. So there are times when it's a very bad thing and it looks like it's all gotten better, and then you're back to it again. And so making changes in the trial can be driven by both internal and external evidence. Internal might be evidence that you've collected within the trial. External would be another trial that's published, or for some reason you're convinced by evidence outside of your own trial that should influence yours. And that certainly happens within the trial environment. A lot of these decisions are driven by uncertainty. So we have a data safety and monitoring board, and ultimately the decisions come down to them. Finally, I'd say, are you prepared for how the findings may be interpreted? We have to preempt both criticism and over-acceptance. As you'll probably be aware, in the beginning, there was an over-acceptance of signals of effect from small trials, for example, of hydroxychloroquine. We're seeing the same thing now with ivermectin. But you can also have over-criticism of interventions because people don't really want to get burned again in the embarrassing way that they were with hydroxychloroquine, for example. And finally, can we influence global guidance? And that's where it goes from us being clinical trialists in general to interacting with guideline committees. And in general, they can be quite heterogeneous in terms of their knowledge of both methodology and also the disease. There's a lot of bad science going on these days from guideline committees and from pharmaceutical industry. So what makes the useful trials in this area different? And Srin's going to talk about a couple of these, REMAP, CAP, and Solidarity. There's another trial called the Recovery that was already discussed. There's the Principal trial where we heard about the budesonide, and then there's Together trial. There might be one or two others, but there really only are a handful of trials that I would say are useful information. And they all have the same phenomenon in common, which is that they are platform trials, they're adaptive platform trials. So I think personally, this is the way for the future of clinical trials. Not everybody will agree with me, but for me, I won't be going back to doing 2- and 3-arm trials. The trial that I'm going to present some results to today comes from Brazil. So I mentioned know your epidemic, which means go after where the disease is. Brazil got hit really badly for a variety of reasons, and they have had their own homegrown variants, as well as they've now experiencing a wave of the Delta variant, and we have about 12 active sites occurring in Brazil within the public health sector. To quickly address this, there is a publication that we provided on our protocol as well as our statistical analysis plan, but for the sake of today, all you really need to know is that we dealt with adults who were positive for COVID and symptomatic within the last 7 days, an average of, I think it was 3 days or 4 days, depending on which analysis that you look at, and over the age of 50 or with a known risk factor for COVID. When we began this, we began it as a 3-arm trial back in June 2020. We looked at hydroxychloroquine, lopinavir, and placebo. We did an interim analysis looking at the data after approximately 200 patients per arm, and we decided to kill the hydroxychloroquine and lopinavir arms because they were not demonstrating any treatment benefits. And I think this is a key aspect of an efficient clinical trial, is looking at your data early and being prepared to stop the treatment arm if it's not giving you any useful information. After 200 patients per arm, you should be able to tell whether or not there's a treatment effect, at least to be likely to be detected in the future. After that, we started a 4-arm trial. We began with fluvoxamine, metformin, and the continuation of placebo. We started this trial with single-day dosing of ivermectin, what I'll refer to as low dose. Ivermectin at that time was actually something that a lot of people in the scientific community were very interested in and open to. There was debate about whether or not the preclinical ivermectin evaluations made sense or not, but everybody felt it should be evaluated because there were small trials coming out of relatively obscure environments, Bangladesh, Iran, Iraq, places you're not used to seeing clinical trials. But they were all very positive. So for that reason, a number of people, myself, the Principal trial, the NIH at the moment, began ivermectin within the platform trials with some optimism. We were then, we did a consultation with those who were supportive of ivermectin. They felt that we were using too short of a duration of dosing with ivermectin, and for that reason, we increased the dose to 3 days worth of dosing. We then did an interim evaluation and discontinued the metformin arm for futility. We then continued on with fluvoxamine and high-dose ivermectin. We introduced peginterferon lambda based on some really encouraging evidence from Jordan Feld in Toronto, and we introduced doxazosin because of some compelling preclinical work out of Johns Hopkins. At the end of July, beginning of August, we did an interim evaluation. We stopped fluvoxamine for superiority over placebo, and we stopped ivermectin because we had hit our intended sample size, and there was no evidence that it was going to show important clinical benefits, so the DSMB recommended discontinuing it at that point. We continued on, and we continue on now with interferon versus placebo. Doxazosin has been paused at the moment because of concerns about harm, and we'll see what the DSMB decides to do with that. It's paused, it hasn't been discontinued just yet. We will likely be entering a new intervention in the next couple of weeks, which is the fluvoxamine in combination with an inhaled steroid. We're waiting on ethics approval for that. Our statistical approach uses a Bayesian model for the primary outcomes because it's probability driven, the likelihood that something is giving you a treatment effect or not, and then a frequentist model for all secondary endpoints. DSMB are the ones who make the decisions on stopping, proceeding, or making any changes. Our primary endpoint was a composite endpoint of admission to hospital care. Now, because of the pandemic in Brazil, there were mobile hospital units were created. We refer to these as COVID-19 emergency settings. And we looked at whether or not you were admitted for retention in there, defined as a participant remaining under observation by a physician for greater than 6 hours. And that doesn't include the wait time. That's whether the physician recommended staying in that mobile unit, or transfer on to a larger hospital. Because of the overcrowding at the hospitals, many patients wouldn't end up making it to the hospital, and so they were retained for several days in the COVID-19 mobile hospitals. This is a photograph of those mobile hospitals, and there's something like you might imagine from a MASH unit, a little bit cleaner, but they became overcrowded as well, as you can see at the lower right-hand slide. So our findings on different interventions, well, initially we published the hydroxychloroquine, lopinavir, and placebo evaluations as one trial, a 3-arm trial, came out in JAMA Network Open. We showed no benefits at all on viral load or on clinical outcomes with those interventions. When we looked at metformin, we stopped it for futility. We found no benefit that was of any use within metformin. I'm presenting here the results from ivermectin that led to the discontinuation of it. There are some updated results that I'm not presenting here. And we found no clinically meaningful benefit to ivermectin, a relative risk of 0.91, confidence interval 0.69 to 1.19, and also no observable clinical importance for mortality benefit. Fluvoxamine, on the other hand, we stopped because of superiority. And this is really quite compelling because there was a smaller study out of the US with Eric Lenzi and Angela Ryerson published in JAMA about a year ago, showing that it had important effects within a small trial, about 80 patients. So we did a randomized trial where we randomized within our design 741 patients to fluvoxamine, 756 to placebo. As per usual for global health trials, we had a little bit more women than we did men in it. And we found that it had a relative risk reduction of about 32%, with confidence intervals on the upper side, 0.89, an absolute risk reduction of about 5%. So really quite important. However, if you look at the primary outcome using per protocol, you actually end up with considerably larger treatment effect. This is only people who were adherent to taking all of their drugs, which in fact was the large majority of the patients. A relative risk of 0.34, so a 66% relative risk reduction. upper confidence intervals that are quite meaningful. And this also played out on mortality. So per protocol mortality was significant. The intention to treat mortality was not significant, but the direction of effect and the effect size was quite similar to the primary endpoint. So the next intervention that we're doing will be fluvoxamine plus an inhaled corticosteroid. With all likelihood, that'll be fluticasone. And we model out the effects of that. If you assume that the treatments are additive, then the 32% reduction in the primary outcome of hospitalization, in addition to the primary outcome, the hospitalization outcome from the principal trial, which was a 26% reduction, even though it wasn't conventionally statistically significant. I'm quite impressed by that trial. It would give us an overall intention to treat relative risk of 0.50 that could be quite meaningful. So we will evaluate that in the next short while. And just before I started here, we found out we are being supported to do some long COVID work as well. So this trial has evaluated 8 different interventions over time. And I think it's a very useful way because we were able to kill 5 of these interventions based on futility. We were able to kill them early. And I think that's the way that we should move forward with clinical trials. So in conclusion, unfortunately, we haven't received anything from Canada on funding this trial. And we don't receive a lot of callbacks when we make calls to the Canadian government for supporting this. We instead received the majority of our funding from a 32-year-old young Irish person named Patrick Collison out of Silicon Valley. He's been very generous, and the rest of it we received from the Rainwater Charitable Foundation. Those are the members of the team. Thank you.
Thanks a lot, Ed. This was very impressive, and I'm looking forward to the discussion. Just reminding everybody, we tend to be delayed, compared with the originally planned schedule. So I'm just handing over to the next speaker, who is Rob Fowler, a professor of critical care medicine at the University of Toronto, the chair of the Canadian Critical Care Trials Group, and you will be talking about the CATCO trial. Handing over to you, Rob.
Thank you so much, Peter, and thanks for the opportunity to participate with so many terrific speakers. I'll present a little bit on the Canadian treatments for COVID-19 trial, and I'll do so on behalf of both myself and Srinivas, who will be speaking afterwards. This is a project that has been supported by a number of organizations in Canada, locally at different hospitals and universities, and importantly so by patients, families, research coordinators, principal investigators across the country. In terms of disclosures, I would say only holding grants from CIHR for this trial and also a COVID-19 network of clinical trials networks, but no personal or professional disclosures from other sources. This is a clinical trial that has been done and conceived in collaboration with the World Health Organization. It's called Canadian Treatments for COVID-19 in Canada, and it's called a Solidarity Trial in most parts of the world outside of Canada. It's focused on inpatients, testing medications, and it's, as Ed and others have mentioned, has a couple of features that are not unique to this pandemic, but certainly have gained a lot of traction during the pandemic in terms of being adaptive, looking at adding and taking away drugs in the context of the normal operations of the trial, dependent upon early signals from the data. And it's done on the background of a platform, a common analytic set of analyses and processes across, in this case, many drugs. And I'll note, thanks to CIHR as being the predominant Canadian funder for this global trial in the Canadian context. The structure is a tight collaboration where this trial operates somewhat independently in Canada, but feeds data to the WHO using common platforms, and in most cases, common case report forms with some ability to collect augmented data in the Canadian context. As a graphic representation of countries across the world that are engaged in this trial, you can see that much of the world has been covered, and the coverage is even a bit greater right now. And I'll go through some of the progress over the course of the trial in the next few minutes. The overall objective is to evaluate the clinical effectiveness of medical interventions compared to the standard of care in patients hospitalized with COVID-19. And a couple of the results have been alluded to by Emily and Ed and others. Primary outcome is mortality in the hospital, and then looking also at clinical efficacy on a number of other metrics, including severity of illness over time, days receiving certain therapies in the hospital, and mortality at different time frames, and safety of medications that have often not been very well studied in advance of the COVID indication for study. Eligibility criteria, adults that have confirmed by some objective mechanism virus in their system, and they're hospitalized at participating centers and not expected to leave those sites to a non-participating site in the short term, that they are expected to survive at least over the day, and of course don't have an allergy to study medications. There are generally very few interventions that had specific exclusions, I would say, and that's a general principle of trying to prioritize medications to study from the perspective of WHO, looking to make this as broadly generalizable across the world, where there's often less opportunity than in high income and well-developed healthcare systems to be looking at drug-drug interactions and to be looking at specific elements of adverse effects or reasons why drugs can't be co-administered. As alluded to, the reason to study many of these medications, some of the rationale has come from science, to be sure. Some of the rationale has come from the need to investigate therapies that were being used on the basis of non-science, but either early preliminary and very, very questionable sort of observational studies, and a lot of political overlay and advocacy for use. In the United States, just to put a point on that, looking at prescriptions of hydroxychloroquine or chloroquine across the period leading up to the spring of 2020 and after, showing an incredible spike in prescriptions as folks were wondering about its effectiveness in preventing or treating COVID-19. No evidence base in March and April to support this, but this is sort of real world response to circumstances around a pandemic. The first medications studied in CATCO and Solidarity included hydroxychloroquine with some background data, certainly mostly in vitro data around lopinavir, ritonavir, traditionally an anti-HIV medication, remdesivir as highlighted earlier, and interferon. And subjects were randomized to receive one of these drugs or standard of care in Canada or in global centers. And I would say from the end of March of 2020 up until the end of the summer of 2020, there was a great uptake around the world and terrific enrollments in Canada and outside of Canada, allowing us to come to some conclusions early in the late fall of 2020. What have we learned? So in brief, from the first phase of this trial, we've learned that lopinavir ritonavir is not an effective therapy, looking at hospital mortality as one of the outcomes here. We would say that hydroxychloroquine, similarly, as alluded to by a number of the co-speakers, does not work, and if anything, could be associated with increased harm in broad scale use. Mechanisms a little bit unclear, may or may not relate to side effects and cardiac complications. Interferon, specifically in the formulation used in CATCO and Solidarity, does not seem to be effective for most hospitalized patients. And I would flag in the formulation used because there are still a number of ongoing investigations with different formulations of interferon that lead us to believe that there may be good reason to imagine differences based on formulation. And as Emily talked about, remdesivir, that is an antiviral, and I'm showing results just of the CATCO Solidarity trial here with really superimposed Kaplan-Meier curves looking at mortality as an outcome. Yet this has been the subject of of still interest, because, you know, looking at a combination of subgroups and thinking about, you know, are there possibly with larger numbers of patients or potentially honing in on different patient populations, the opportunity for effectiveness medication, enough interest globally that WHO continued to enroll patients in this trial beyond release of the first phase results. And in Canada, we continue to enroll patients even beyond the WHO closing of recruitment. And those results are almost ready for dissemination for peer review and then eventually public review and impact. Where are we now? We have the second main global solidarity manuscript in preparation. And in parallel, there is a Canadian-centered manuscript in preparation that will focus upon the Canadian experience with remdesivir. And hopefully, I would say within the next month or two, those results will be widely available. The next phase of medications are under study. We have over 60 sites across Canada. Some of the sites here are in early phases and are not yet able to have a research team in place to randomize, but we're working actively, particularly in sort of north and rural communities, to try to augment that capacity and allow a combination of observational and hopefully experimental research to occur. We've got about 1,500 patients enrolled so far in the trial, and globally there's moving in on 15,000 across many countries, many hospitals. A very nice collaboration with Canada, the WHO, and many member states across the world. The medications that are being tested right now, and I've listed them as the next phase, include artesunate, imatinib, and infliximab as a first and then an interesting, I would say, twist on the next phase of CATCO is that we are incorporating another Canadian trial looking at the impact of losartan and angiotensin receptor blocker in the treatment of patients with COVID-19. And this is an incorporation of a pre-existing and CIHR funded trial into CATCO in order to help augment enrollments across CATCO sites. And then a third randomization for patients that are still sick and remain on oxygen at day 10 to have an extended course of dexamethasone on top of the currently recommended treatment course. And so a twist into the next phase in conjunction with WHO, but with Canadian specific additions. Some of the challenges that we've experienced along the way with this trial have been not, I would say, isolated to this trial, but are ones that are probably generalizable across the Canadian experience, and that it takes a while to get things up and running. This is a sort of research time in motion study that is in preliminary form, done by a number of collaborators within the CATCO team, and looks at different milestones in terms of protocol, showing up at sites, REB submission to REB approval, frankly, very quick in our experience, contract submissions and executions taking longer, and then the stage of contracts being executed and to getting a patient enrolled, getting all of the startup stuff done, getting a team in place, going through a number of other sort of regulatory hoops takes a long time between REB approval and first patient enrolled. And presenting here sort of median times, we have very short times at one end of the spectrum and long times at the other, but a median time that reflects an important metric on efficiency of our research in Canada, still not where we need it to be. Well, what are we working on? We're working on trying to get ourselves looking a little bit more like a national healthcare system when it comes to research. We have pointed to the UK example commonly in Canada as something to replicate in terms of having more national sort of one-stop approvals for trials from the research ethics perspective, from the regulatory perspective, and from the contracting perspective within our hospitals. Challenging based upon the way we administer healthcare in the country that is provincially, territorially administered, and in some cases federally administered, but not having the necessary contacts to smooth out those interprovincial and territorial boundaries with respect to both ethics and in particular, I would say, contracts and some of the regulatory components. And so this is a challenge for us to work on collectively over the next year so that we're better positioned for the next challenge. On behalf of both Srin and myself, thank you for the opportunity to go over CATCO, and thank you so much to all of those that are participating, collaborating, and thank you to CIHR and to sponsors in Canada. And I'll stop sharing there. Thank
you very much, Rob. I'll just rush over and hand the mic over to Ryan Zarychanski. You're an associate professor at the University of Manitoba, and we've worked now for quite a while, together on therapeutic dose anticoagulation using heparin. Looking forward to the overview related to your multi-platform trial, Ryan. We can't hear you yet. Still not, or at least I can't. Yeah, now it works. All good.
Okay, sorry about that. I was pressing the wrong button vigorously though. Okay, I thank you for this invitation. Quickly, we'll talk about therapeutic heparin in COVID-19. I have disclosures that are related to grant support with regard to the study of heparin in COVID. I'm going to go a little bit quick, conscious that we're short of time. So early in the pandemic, there was a lot of blood clots associated with COVID-19 that were reported, and the incidence of blood clots varied from like 5 to 30%. I think they're overestimates. There are venous and arterial events both reported. I think it was a larger problem than it occurs currently because of changes in how we care for the patients and the treatments we use. That said, poor outcomes are still associated with COVID-19, regardless of the estimates that may be not as generous as they are on the slide here. And so there's this concept of immunothrombosis, so the immune system perpetuating blood clots, whereby immune system helps to inactivate pathogens, but also activation of neutrophils or monocytes can also cause inflammation and activate coagulation, causing large blood clots or small ones. And we know that people with respiratory failure in COVID have a degree of not only little bits of hemorrhage in their lungs, but small blood clots as well that probably contribute to organ dysfunction and morbidity and perhaps a bit of mortality. There's also this, just like there was enthusiasm about hydroxychloroquine at the start without any real data, there was total enthusiasm about D-dimer that it would somehow identify a patient population, that could benefit from some unknown treatment that we did not have yet. And so it was recommended that we measure D-dimer in all hospitalized patients for reasons that totally aren't clear because we didn't know what to do with the information. Although it probably does have prognostic significance, that is, it probably does identify a patient population that is sicker if the D-dimer is higher, and that's a measure of clot breakdown. So they have to have some degree of inflammation or blood clots to have a D-dimer. And so we wondered really early about heparin because there was some blood clots reported, lots of inflammation going on. Inflammation and blood clots conspire to cause organ dysfunction. And we thought, well, what about heparin? Heparin is a really inexpensive, widely available, familiar drug used all over the globe, that it's a good blood thinner. And in addition to being a good blood thinner, it is a molecule with known anti-inflammatory properties. And it also has antiviral effects that we don't know how clinically relevant they are, but the molecule does inhibit the spike protein and may reduce viral entry into the host. And so we wondered if, with therapeutic heparin could improve outcomes in patients with COVID-19 or are hospitalized. And this hypothesis was shared amongst 3 platforms, and platforms are independent trial units. And so what we did is that it was a bit of an organic experience, but at the end of the day, what we did is we had 3 independent platforms, REMAP, ATTACK, and ACTIVE, and they're on different parts of the world, some overlap. 10 countries, 400 sites, where we all work together to harmonize all our protocols into one single trial protocol. We administered the trial separately, that is with different regulatory environments, but we had a single protocol, single set of core outcomes, and an analysis plan that was line, patient line data, so not a meta-analysis. And we had a DSMB oversight plan where all the DSMBs agreed on decisions with regard to stopping for different trial decisions met. This was a Bayesian platform, Bayesian framework as well that Dr. Mills referred to in the types of analysis that could be done. And so we included people that were hospitalized with COVID, and we wanted to know, does therapeutic anticoagulation work differently, or does it work at all? And we wanted to know, does it work in severely ill patients or moderately ill patients? And in the moderate ill patients, we also wanted to test this D-dimer business going on to bring some sanity into how we perhaps use D-dimer to guide treatment decisions. And so we defined severe disease by those who need respiratory or cardiovascular organ support. And so you have to be on high flow nasal oxygen or worse, like non-invasive ventilation or invasive ventilation, or needing blood pressure medications to keep your blood pressure up in an ICU primarily. One of the platforms did not need the ICU requirement. And the moderate disease was defined as people are hospitalized who didn't need this respiratory or cardiovascular organ support. Okay, so it was all defined by organ support. And we excluded people who were admitted for more than 3 days, or for the most part, and or expect to have short admissions or other contraindications to anticoagulation. Really sensible stuff. And the intervention was heparin, so it was low molecular weight or unfractionated heparin. Those are just like different permutations of the same kind of molecular structure. And we used it in really common doses for the treatment of blood clots, even though these patients didn't have recognized blood clots at the time of enrollment. And the control was whatever we normally give to prevent blood clots in sick hospitalized patients. So it's usually a lower dose of that medication. And we gave it for up to 14 days or until hospital discharge. The primary outcome was an outcome that we called and defined as organ support free days. So organ support free days is not just organ support, it's mortality plus your requirement for ICU level care or organ support. And if you died, you got a low number assigned -1. And if you never needed organ support, you were assigned a value of 22. And if you survived and needed some sort of organ support in there, you got a number. So if you were survived and had 16 days on a ventilator and then were off, then your organ support free days would be 5. Okay. And this reflects both mortality and morbidity. It reflects something that's important to patients and to health systems. A higher number that I'll present and based on an odds ratio is a better outcome. And this outcome was chosen because it can work across a spectrum of severity of illness. So moderate or severe could still be interpreted with organ support free days. Although in the moderate group, it functions as a little bit more dichotomously, like, do I ever need organ support or not? Because most people don't on the ward. And in the ICU, it functions more like, it's kind of like a continuous measure at some level, but it's not, it's an ordinal scheme. Analyzed as such. Anyway, so secondary outcomes are components of the primary outcome. Clots, because we wanted to report on clots. A lot of people were interested in it. Bleeding and complications of heparin, the main one being heparin-induced thrombocytopenia. Okay, so we had 3 independent platforms working together to achieve a goal as quickly as we can in something that we called the multi-platform trial. It was a Bayesian trial with frequent looks at the data, about almost monthly, approximately monthly. And we had different categories or groups that we could stop the trial in. We were really interested in severe and moderately ill patients. And within moderately ill, we wanted to know about the high or low D-dimer strata or subgroups on baseline. So we enrolled about just over 2,200 patients in the moderate group, just over 1,100 in the severely ill group at baseline, So this is just a mix of who they were, age, sex. Most people were on just regular oxygen in the moderately ill. A couple were on high flow or non-invasive because in one of the platforms in REMAP, you had to also be in an ICU setting. And then in the critically ill patients, most people were on high flow or invasive or non-invasive ventilation. And so what we found out was that therapeutic heparin, affordable, accessible, familiar drug, is of no benefit in critically ill patients. In fact, when given empirically at baseline for those on organ support, there's a high probability of harm. So we should stick with what we normally do, which is prophylactic doses in that group. But in moderately ill patients, so those who are not yet that sick, therapeutic heparin was a benefit. This is like a horizontal box plot, or heat, it's a kind of a heat map almost horizontally that shows that red is dead, the patient died, and dark blue is never needing organ support. So you can see that more people died in therapeutic heparin compared to usual care, and the opposite is true here. So a little more people survived in therapeutic heparin and moderately ill, and there's more dark blue shading, more dark blue of a shading, more benefit. And so these are adjusted odds ratios, adjusted for important covariates like age, sex, time, site. And so how do you relate that to at the bedside more easily? And so there's a 4% adjusted difference compared to baseline risk of requiring organ support or dying if you were not on therapeutic heparin. So there was about almost actually 4.5% increase in you or benefit from you of a patient not dying or needing organ support. And so what about D-dimer? So that was the hypothesis, like, does D-dimer make a difference? No. And so the relative benefit of therapeutic heparin was consistent in all D-dimer groups at baseline. D-dimer groups were defined as 2 times the upper limit of normal or below for high and for low. But the absolute risks are different according to D-dimer status. And so are the absolute benefits. So the relative effects are the same. Absolutely, there's a greater benefit because of the higher event rate in patients with higher D-dimer. So it does have prognostic significance. Secondary outcomes in severe, I mean, none of them are that, like they're not positive in a sense, like they're not, there's no benefit of heparin. And so, when we look at, the organ support free days that I mentioned, there's no benefit from heparin. Yes, you can reduce thrombosis a bit, 10.4% to 6.4% in the unadjusted proportions, but when mortality is mixed into that, there's no difference in major thrombotic events or death. So really no clinically meaningful benefit. There's a bit more bleeding, but bleeding rates were low, 3.8 compared to 2.3%. In moderately ill patients, there's consistent benefits with therapeutic heparin. So I talked about that, 4.5% absolute adjusted increase in survival without needing organ support. And so it's like, 25% less of the patients will go to the ICU or require ICU level care in a hospital, which I think is a really meaningful endpoint. But there's also less progression to intubation or death. There's less, there's a bit less thrombosis, but thrombosis wasn't a big player. And when death is in, there's a slightly reduced incidence of thrombosis and death as well. Bleeding, like major bleeding, was low, 1.9 compared to 0.9, so a small increase, but low overall. So therapeutic heparin reduces organ support and death. But does it decrease, respiratory organ support or death? Yes. Does it increase just intubation and death? Yes. It's a smaller cohort each time you cut the pie that way, but the results are consistent along that spectrum. And so in summary, critically ill patients with COVID-19 are admitted to hospital are not benefited by therapeutic heparin up front. But in critically ill patients, there's a high probability of benefit, like 99% certain benefit. And in the subgroups there, you'll see 99% organ support free days to 21, but also less progression to intubation and death, less thrombotic events as well, and low rates of bleeding. What about other trials? Well, the results are coming together in a picture of consistency, and these are the results from the rapid COAG trial that Peter is involved in there as a co-PI. 465 hospitalized patients with moderately ill COVID-19 and elevated D-dimers. The results are eerily similar, like the protocol is similar, and the results are eerily similar to the MP or the multi-platform trial I presented in terms of organ support-free days, intubation, mortality, low rates of bleeding, all in the same direction for the most part, really consistent in their magnitude and in the direction of benefit as well. So you can see that this is like a frequentist meta-analysis combining the results of both trials. And even when we look at organ support-free days and as defined in the multi-platform trial, the results are consistently beneficial, that is, for therapeutic heparin in moderately ill patients. And then the results are consistent in critical illness too. So this is a trial of higher dose heparin called the Inspiration Trial in JAMA in critically ill patients that was no benefit. And so it wasn't quite the therapeutic dose, but it was higher than standard, and there was no benefit in critically ill patients. And again, there's another trial called ACTION, but they studied a NOAC or an oral kind of blood thinner, and there was no benefit in moderately ill patients. But that's not contradictory. It's a totally different drug. It's oral. It's not the same mechanism of action as heparin. Doesn't have the same anti-inflammatory, antiviral, potentially antiviral effects, and so it was not beneficial in a stable MORD-based population. And again, consistently in the unstable critically ill patients, enoxaparin at a higher to low, which is a type of heparin, was not beneficial. So again, more evidence that therapeutic heparin empirically in critically ill patients is not beneficial. There is another NOAC, another novel-ish oral anticoagulant being studied primarily in Brazil and Spain that is ongoing, and they've got 3,000 patients enrolled. So we'll see if none of the NOACs work, or if it was molecule specific. That is, the effect could be molecule specific. So we'll know about that in some amount of upcoming months. There are a swarm of meta-analyses coming, like so many you couldn't even fit on the slide. Every week I get asked to give the data around meta-analyses. Today is no exception. And so there is like standard meta-analyses coming, network meta-analyses, individual patient meta-analyses, living systematic reviews, all evaluating anticoagulation heparin. And there's some really clever ones being done by the WHO and individual patient meta-analysis that our team is doing that I think will help pull together all the data, because I presented about 4 trials, but there's about 20 ongoing. And like Dr. Mills said, majority of these are too small to really make meaningful impacts. And so we'll have to figure out what to do with the totality of data. So the recommendations: therapeutic heparin in hospitalized non-critical ill patients is recommended for up to 14 days or until hospital discharge. A NOAC is not recommended. The role of D-dimer is unclear, may have prognostic significance, and use whatever standard dose in critically ill patients. Just want to thank everyone. This is a kind of a global trial, over 10 countries, almost 400 sites enrolling. Those are the 2 first authors and co-PIs. Many people were involved. That's Ewan Gallagher on the left, Patrick Lawler on the right from Toronto. And I represent just a large team, and thank you for the invitation to be here.
Thanks a lot, Ryan. You just gave me the opportunity to just smoothly transition to Srin in this case, talking about the prospective meta-analysis that you initiated, Srin, on behalf of the WHO. So just handing over to you that we still have enough time for a few questions in the end.
Sure, I shouldn't take that much time. I just want to give a high-level overview of the things we're doing at the level of the WHO. This is as part of something called the REACT approach, so the rapid evidence appraisal for COVID-19 therapies. I'm from the University of British Columbia. Some disclosures here: I'm not speaking on behalf of the WHO. I'm a consultant for them, and I work sort of side by side by them, but I am not a direct employee. I receive salary support from those organizations, public funding from CIHR for COVID trials, and I do chair the clinical reference group for the province of British Columbia for COVID-19. Back in February 2020, we came up, a few people, about 100 researchers, went to Geneva to define sort of the global research roadmap for what we called then 2019 novel coronavirus. This was before COVID-19 even was named accordingly. And from a therapeutics landscape perspective, the first objective was to get a vaccine. The second objective was to make clinical outcomes better. And the first few agents, antiviral agents, immunomodulatory agents, particularly steroids, and supportive care interventions through high quality randomized evidence. The challenge then became that, as everyone has alluded to earlier, that the trials were, one, too small, and then two, too many, in the sense that, say, hydroxychloroquine, which is the example everyone gives, there was hundreds of trials that started within the first few weeks of this pandemic, and similarly to convalescent plasma, ivermectin, Kaletra, and a number of other drugs. Many trials looking at the same question, or at least similar questions. And then you have patients in China. This is a picture from a Wuhan hospital where they didn't have high-level evidence to define the therapies. And so you're balanced with a world where you have lots of trials starting, and you have patients who need high-quality evidence to define what to give them. And so in that landscape, the WHO struggled to figure out exactly how to provide guidance and evidence to make sure patients got the highest quality care available. The challenge is that this is sort of a Goldilocks problem, in that you have a few giant trials based on sort of the infrastructure of the NHS and NIHR, say recovery, solidarity with the convening and collaborating power that the WHO offers, many medium-sized trials, REMAP-CAP, active, together, and so on, and way too many tiny trials. Many of these things asking similar questions. And so if you look at hydroxychloroquine, for example, recovery, solidarity, REMAP-CAP, active, and then many of the tiny trials all were asking the same questions about whether hydroxychloroquine worked. And so the challenge in that context from a guidance perspective is to synthesize that evidence quickly and produce usable guidance for the world for clinical practice. One approach is this, and this was a sort of a schematic that was reported in JAMA about direct pooling of individual level data of patients as it accrues during clinical trial conduct. And so a group in New York did this for convalescent plasma, or at least started to do this, whereby each trial would send their individual patient data to a central body, and then there would be a meta DSMB that would review this data and then report back to each of the individual DSMBs as to what they should do with the data as it accrues, and when benefit or harm or futility was achieved, all of the trials would stop. The challenges with this meta trial approach are legion, and I think from a legal perspective, there becomes that challenge of actually working through the contracts in very quick timescales. Trialists have, and I put my picture there, for example, trialists have sort of some personal issues about sharing their data in that sort of rapid timescale and integrity of the trial, particularly harmonizing protocols, which oftentimes leads to challenges in interpretation. And then funders want to see their individual trial get reported all the way out to individual completion. And so we look to the Cochrane group, and they have a specific approach about prospective approaches to accumulating evidence. And many of these authors have been involved with our processes for this prospective meta-analyses approach that the WHO is spearheading. And so what we decided to do back in, say, April of last year is to figure out what treatments would be amenable to this approach, find all of those trials, write a protocol with the trialists for a thoughtful, pre-specified meta-analysis, time things very carefully with those individual trials, and then build close relationships with journals and those trialists to make sure that it all happens in a collaborative, cohesive way to produce high quality evidence for the world. And so when Recovery reported their dexamethasone results, this is their peer-reviewed manuscript, but the day they got their unblinding to their principal investigators, and so Peter Horby and Martin Landry, we were on a telephone call with them, working through the processes as to how to get this data out to the world in a synthesized way. given that we've already had a close relationship with them before they reported their results for steroids specifically, given that that was one of the priority conversations and questions out there in the world. Particularly with this sort of like reasonably, one could say antiquated, one could say conservative approach about how do people change practice, and whether or not people can change practice with just a single trial, or whether they need a combination of data sources to actually impact clinical practice. And so what we've done is, this is a paper from June or August of last year, and Peter was involved with this as well, about a meta-analysis of steroids, whereby what we did was pool all of the data of the ongoing trials, report them all simultaneously in a pre-specified way with the journal itself, and produce high-quality evidence as to the role of steroids, acknowledging that the data was primarily driven by one large trial, but having many medium trials report things in cohesion with this is going to be useful for clinicians and guidance panels to produce high-quality guidance. Similarly, with the IL-6 blockers, a larger number of trials in this context. This is tocilizumab and sarilumab specifically, working closely with the trialists and with a pre-specified protocol, coming up with a sort of a very clean answer as to whether these drugs work in patients, and looking at specific subgroups and specific outcomes that would be impacted by the availability of these drugs. And so it's a pre-specified process that has to be very well coordinated, and the WHO is great at that coordination process. And so these two meta-analyses led to the only two strong recommendations that exist for treatments for COVID-19 in any setting, outpatient or inpatient, specifically corticosteroids and IL-6 blockers as strong recommendations. And the challenge with sort of IL-6 blockers is that these recommendations lead specifically to a large number of conversations about accessibility and cost. And with sort of recommendations coming from groups like the WHO, a lot of sort of access provisions come into play. And so the strength of this cannot be understated. Next is anticoagulation. I put a data sharing is caring slide here, and many of us were on a call just before this conversation here about processes of an anticoagulation perspective meta-analysis to very cleanly define the population that would benefit and how much benefit they would have, so that guidance can be offered for the world, and then accessibility of the products can be worked on. I'm not going to get into sort of large trials versus meta-analyses of smaller trials from a methodology perspective. This is a paper from John Ioannidis back in the 90s, but we don't have time to discuss that in detail. So from too small to many trials, we still have too small and too many trials, and the very small trials aren't probably going to lead to much value, but a lot of the medium-sized trials and the larger trials have led to this unprecedented global collaboration. I want to acknowledge a handful of people: John Marshall at St. Mike's in Toronto, Claire Vale at UCL in London, statistician Jonathan Stern from Bristol. And I also want to highlight Janet Diaz, who's the lead for health emergencies for clinical work for COVID-19 at the WHO. She doesn't get as much of the attention as many other WHO colleagues do, but her work has been tireless to help define clinical management around the world, and she deserves a lot of credit for a lot of this work and a lot of other work for COVID-19 management around the world. So to conclude, prospective meta-analyses are a useful adjunct when multiple trials are ongoing during a public health emergency, but it does require this collaboration among trialists in an unprecedented way. I'll stop there and leave a few minutes for questions.
Thanks a lot, Srin. So after this overview, first story that I keep wondering, and it's really a question just to all of us. When I teach clinical epi and trials to my students, I always tell them be really skeptical about subgroup analysis. But the story here, and several of you have just pointed that out, it seems that for COVID-19, we have two watershed moments. One is whether I need oxygen or not, and the other one, whether I need formal organ support or mechanical ventilation or not. And this seems to be relatively strong effect modifiers that are also confusing matters sometimes in guidelines when I look at some of the, the garden variety meta-analysis out there as well. Would any of you comment on that? Am I too much of a clinician now, or do you agree with me that this disease behaves a bit differently than what most of us are accustomed to in other conditions? Not sure, Ryan, perhaps you first. Were you surprised about the results that you found in your 2 multi-platform trials?
Well, I mean, yes. I think it shows why we do trials, though. I mean, if we knew, we were bad at guessing. In the States had to give 100,000 doses of plasma to realize they weren't giving out effectively. Hydroxychloroquine was billed as a game changer, and et cetera, et cetera. And this is why we do it. And so I thought, and many of us thought, that therapeutic heparin would benefit mostly the critically ill patients. And we were totally wrong. So whatever I say from now on should be with a grain of salt, because I clearly don't know what I'm talking about. But this is why we didn't have to do trials, because we're bad at guessing in medicine. And these are the only way to bring about a structured, reproducible, transparent, informative way to conduct clinical care. And so, yes, we're surprised. And we have learned in the pandemic that timing is important with regard to different treatments. We know this in steroids. We know that the effects tocilizumab can vary. We clearly know that we know that in heparin now that there's a patient population that benefits and those that doesn't. We're starting to understand that not all convalescent plasma is probably null, and there may be a patient population, whether it's immunosuppressed or otherwise, that could benefit from that as well. And so, yes, we are learning about differential treatment effects by a host of variables that include virus, host, and timing related matters.
Thanks. Ed, you're one of those trials that uses a placebo. A, should we use placebos more often? I also think of Emily's comments. And B, how did you actually set up this placebo control? What kind of placebo were you using here? Because you have a great many different interventions that you're testing.
Sure. So those are good questions. There's a lot of decent debate in the scientific literature about the role of placebo in different trials. One of the analyses that we will always run within our trial is, let me explain how we do placebo within our trial. Is we only use the concurrent controls in our trial. As you saw when we're going forward, we had a placebo group when we did hydroxychloroquine, but once we stopped hydroxychloroquine, we're no longer using that placebo group or control group. We don't use historical controls. There's some debate about how that should be done. We happen to not. So we're just using concurrent controls. Reason for that is that hospitalization rates were around 5% at the beginning of the pandemic. In Brazil, they're around 15% now. So the control group has changed in important ways. Within our trial, you get proportionately randomized to the placebo group, the matching placebo of the intervention, according to the number of active interventions that there are at any time. So if there's 4 arms in a trial and one of them is controls, then you have a 1 in 3 chance of being allocated to a matched placebo to whatever the interventions are at any time. And we run an analysis to see whether or not any of the placebo groups exhibit a different treatment effect. And thankfully they don't. I think at the end of this, it's going to be a very useful trial because essentially we'll have an adjustable head-to-head analysis of 8 different placebos that have been used in the same disease for the same population. So I think it'll be very useful analysis, kind of historically in the end. Thanks.
Thanks. Rob, you had a comment.
I wanted to pick up on your comment about subgroup analyses, Peter. And I think that in the context of the pandemic, where so many treatments were pushed into evaluation at a very, very early stage in the way we would usually think about investigating sort of biological plausibilities and sort of clinical signals, that many of the first trials, even the larger ones, would probably have, certainly benefited from a lot more considered approach and maybe targeting a population a little bit more. And I think in some of the interventions, maybe particularly around the timing of administration, potentially around the patient population in terms of severity of illness, but things that might be more effective early on and then later in the disease course probably really don't have a great hope at helping. I think some of those subgroup analyses, particularly for COVID when things were pushed early into evaluation, might be more helpful than they are for what I would say are sort of traditional time course. And yeah, that was my comment. Thank you.
Thank you. I need to come to a last question, which I would like to ask to you, Emily. What do you think right now? Are we doing a good job with our, approach of adopting new intervention or rejecting them? Are we too conservative? Are we too optimistic sometimes? What is your experience, and how confident do you feel right now with treating patients with COVID-19 compared with 9 months ago?
I think it's a world of change compared to, when the pandemic started. I think a lot of us feel that we have much more randomized control trial data to support different treatments for patients that patients were receiving, but perhaps differentially, just at the level of the clinician who may or may not have given a certain medication. A lot of these treatments were being given from the beginning, and we had no idea who would benefit and who would harm. And now I think we have a much greater idea of that. I don't think that we're too stringent. I think that we need to remove some of the barriers so that we can complete these trials more quickly. But I don't think the answer is to lower our standard for the quality of data that we're using. Yeah, I think we all feel more confident with our treatments now, that we are doing a good job, but we should continue to request the same level of quality of data for treatments.
Thanks a lot. I feel very sorry I need to come to an end here. We're already just slightly over time. Thanks a lot for the excellent presentations to all of you. I really enjoyed just, having a bit of a closer look in some of the trials and, in the backbones of the trials and the mechanics. Looking forward to, hearing more about the findings that are forthcoming, and looking forward to our prospective meta-analysis on heparin, Srin. This could actually impact as well on our treatment guidelines. Handing over again to Rosa, who is basically just finishing this session on behalf of CanCOVID.
Thanks, Peter. On behalf of CanCOVID's team and also Susan Law, the Managing Director for CanCOVID, I would just like to extend our thanks to all our speakers for taking the time to present on such an important topic. And as Peter said, I'm also looking forward to some of the findings. And of course, also thanks to all of our attendees. And a special thanks to Dr. Peter Jüni for the excellent moderation. Just a few announcements that we'll share before we conclude. We do have 2 CanCOVID events next week. One is our weekly speaker series, which is on Tuesday from 4 to 4:30 Eastern Daylight Time, where we will hear from Drs. Sherry Price and Kim McMillan on nurses' experience with mental health pre and post pandemic. And also a second CanCOVID event is with the the COVID-19 Immunity Task Force and CanCOVID workshop, which is on Wednesday, September 29th from 1 to 2:30 Eastern Daylight Time, and that will be on risks and impacts of COVID-19 pandemic on Canada's kids, their parents, and teachers. So some of the latest research results and policy implications. So this concludes our session. Thanks again, and hope you guys all have a great rest of your day. Thanks.
Thanks a lot.
We would like to thank our speakers and members of our network for their continued support and participation in Canada's pandemic response. If you are interested in learning more about CanCOVID or joining our Pan-Canadian COVID-19 Research Network, please visit our website at www.cancovid.ca.